Hello, peoples. Um, I'm sorry I haven't been recording, a lot for a while. And now I'm going to be recording a lot more videos. About 3 videos per day. So, I'm starting with a game called Cell Lab. Go into the tutorial 1. Welcome to Cell Lab. 2 tutorials are provided to make you familiar with our lab equipment. Safety procedures will be taught another day. This first tutorial will teach you how to use our microscope. Please start by moving to the microscope. Okay, microscope. Good job. What do you see here is a petri dish we have prepared for you. To zoom with this microscope, do an inverse pinch with 2 fingers. Please increase the magnification to 300 times. Okay. Navigation. Great. Now move the substrate so that we know we are looking at the brighter area at the top of the petri dish, where the sunlight is visible. Zoom so that this camera covers the screen. Or this area covers the screen. Swipe with 1 finger to move the substrate. Awesome. Micropipette. I see you've used a microscope before. This microscope is equipped with a micro manipulator. One of its uses is to insert cells from our genetics lab on the substrate. Can you insert one of our recently developed cells in the area with sunlight? Tap where you want to insert the cell. Okay. Temperature. Not too hard. Our microscope also has a system for controlling the temperature of the substrate. Turn up the temperature to the observe setting where the cells live, but move slowly. The temperature setting is chosen by the selector in the top right. This thing. Yeah. Observe. Awesome. Incubation. Now you should see the cells multiplying, otherwise insert a couple more in the bright area. We want to incubate the substrate for a while so our cells attain equilibrium. Increase the temperature to the incubate setting. Awesome. Cell tracking. Now you should have a lot of cells. This makes it hard to keep track of them, but luckily the microscope has a visual tracker. The tracker overlays a marker on a selected cell and also moves the substrate to keep it in view. Set temperature to observe again and tap on a cell to tell the microscope to track it. This one. DNA extraction. Sometimes we bring in cells from nature or our noses to study. We can then extract their DNA using the micropipette to study it in the genome editor. Let's do that on one of the cells on the substrate right now. We put a mutagen in the substrate, so some cells should have mutated. Pick an interesting one and tell the tracker to follow it. Please, freeze the substrate before doing this. This one. DNA extraction, good job. Next step is to go to the genome editor, tap genome, and press load. There, choose load from microscope, and the micropipette will automatically extract the DNA of the cell in the tracker. Sequence it, and send it in digitized form to the genome editor. God. Load for microscope. Okay. Sterilization. Great. Now we have the genome of the mutated cell in the genome editor. Your last task is to sterilize the substrate with the built-in autoclave to prepare for the next experiment. This is done using the reset button at the top, a petri dish with an orange arrow. Good luck and keep your fingers out of the autoclave. Congratulations! Now you have now completed the microscope training program. Next up is the genome editor training program. Press the back arrow to exit this first tutorial. Yeah. Tutorial 2, Introduction to the Genome Editor. Yeah. Welcome, this tutorial will guide you through how to use the genome editor. Please start by going to the genome editing lab. This thing, right? Okay, overview. Good! What you see here is the genome editor. It is divided in two parts, the preview window and the mode editor. We will start off by introducing the preview window. It shows you your organism at a certain age. The slider right below the preview is used to set at what age you would like to view your organism. Now set the preview to show your organism at age 2 hours. Okay. Cell modes. Good job. That is all we need to know about the preview for now. Now some genetics. All cells in the human body have the same DNA, but muscle and brain cells behave very differently. This is referred to as different modes of a cell in our lab. The genome editor can handle 40 different modes. Each mode has its own priorities. One mode is edited at a time in the genome editor, and to change what mode is currently being edited, we use the selector edit mode. Now let's edit the node called M5 mode. Okay, initial mode. Now all properties you see below the selector you just used describe properties of mode M5. You might wonder what mode are the cells in the preview in. They are in mode M1 because M1 is by default the initial mode. What mode is initial just tells the preview what mode to start with. Now take M5 initial instead. Now make M5 initial instead by pressing the radio button initial. Property descriptions. Nice work. There are a lot of different properties in each mode. More than we can go through in this tutorial. This means you will have to explore them yourself. If you want more information about any setting in the genome editor, then just click on the accompanying text with an underline. Now read the text about the property red color. You might have to scroll down to see it. Change the amount of red in the color of cells in this mode. Note that this does not affect the behavior of your cells in any way. Finding properties. Good job. Now find 2 more properties and make your mode M5 cells completely red. Okay, child modes. Very beautiful. If you drag the preview time slider back and forth, you see your cells dividing. Many settings control how the divisions take place. Two important settings are what modes the two daughter cells are in. These settings are labeled Child 1 Mode and Child 2 Mode. Set them so that when a mode M5 cell splits, child 1 is of mode M6, child 2 is of mode M7. Okay. Grandchild modes. Good job. You should now see two different cells, one in M6 and one in M7. Increase the preview time to 4 hours to see what these cells split into. Okay. Close the circle. Good job. You should now see 4 cells. 2 in mode M6 on the left and 2 in mode M7 on the right. The family tree looks like that. Now change the family tree into that. Okay, by setting the child 1 mode equals M5 and child 2 mode equals M5 of mode 6 and M7. Tip, instead of changing the selector edit mode, you can tap on cells in the preview to change what mode to edit, okay? Insert into microscope. Good job! Now your initial M5 cell splits into an M6 and an M7 cell. They will further split into 2 M5 cells each, and the life cycle is complete. Now place one of these cells in the microscope and see how you get 3 types of cells, but all with the same genomes. Congratulations! Excellent work! You have now finished the Genome Editor tutorial, and you are certified to work in our lab. Good luck with your research. Oh my god, it works. Yeah. Okay. Bye-bye. Okay, it's time for this level. Level 1, Algae. Challenge not completed, difficulty undergrad. Unlocks gene phagokite and further challenges. Okay. Use the genome editor to make a species that thrives in the environment of this challenge. When ready, put a cell on the substrate under the microscope and unfreeze it to see if it is viable. 100 cells are needed to complete this challenge. Okay. Oh. I did it. That was pretty easy. Okay. I think I will do macrophages now. Another cell type is now available, the phagokite. It can eat the small brown dots you might have seen appear when cells die. Eating these pieces of nutrients makes them grow. No light source is present in this challenge, but pieces of nutrients are being sprinkled on the substrate. Can you make a species multiplied at 300 cells? Maybe. Easy. Okay. Okay. Tutorial 3. Welcome to the Genetic Design tutorial. This tutorial will guide you through how to design a basic swimming and reproducing organism. First, go to the genome editor and make sure that mode M1 is of type phagic height and makes adhesin when it splits. Then set the preview time to 0.8 hours. Nice work. Remember that you can press the make adhesin text to understand what that property is. Now you should see two identical cells attached to each other. They are both in mode M1 since the initial cell in mode M1 currently splits into two cells of the same mode. Now we want to modify this so that the left cell is in mode M2 and the right cell is in mode M3. We do this by editing mode M1. Set child 1 to mode to M2. Set child 2 mode to M3 here. Okay. Setting cell types of generation 1. Excellent, now you should see two cells, different colors. We want the left cell to propel our organism and the right cell to provide it with energy. Make sure mode M3 is of phagic height, then edit mode M2 and make it a flagellic height. Okay. Stop propulsion cell from splitting. Great, you now have a swimmer. You can try that it actually swims in the microscope. It cannot reproduce yet, though. We want the phagic height in the front to split off a spore that can develop into a new swimmer. We thus do not want the flagellic height to ever split, so go to mode M2 and set the split mass to max. So we'll never split. Close the circle. Great, now you can turn up the preview time to 2.0 hours. We see a spore splits off from the mode M3 cell. We want this spore to be of the initial mode M1, so the cycle is closed. Edit mode M3 and set child 2 to be of mode M1. Save your work. Perfect. Your swimmer should be done now. You can try it out in the microscope. It is a good idea to save this genome for future use in the challenges. I think I'm done here. Okay, Macrophages 3. Difficulty undergrad. Unlocks gene lipokite and further challenges. Okay. We are going to try to use your new cell, the flagellic for this challenge. This cell cannot gain energy from the environment, but it can swim. Note that this cell immediately dies if attached to another cell, such that the flagellum is obstructed. The viscosity of this substrate is higher than the previous Macrophages challenge. Can you nevertheless make a species multiply to 420 cells? Maybe. Let's try it. Test lighting quickly, and finished. Alrighty. And that'll do it for today. Goodbye.